Good morning. This is Dr. K. Balachandra, Head of the Department Microbiology. Today I am going to discuss about the most important topic in the microbiology, that is staining technique. Microbiology is the basic branch of science which deals with the microorganisms. We can't visualize these microorganisms with our naked eye. Only with the help of microscope we can visualize the microorganisms. Let us know about different types of shapes of bacteria. Bacteria exists in different shapes like cocci, bacilli, like streptococci, lactobacilli. Those are the shapes of bacteria. To know about the morphology of the bacteria, must and should, staining technique is one of the important tool in the microbiology laboratory. There are different types of staining techniques. Especially most of the important staining techniques is, one is the simple staining and second one is the gram staining. The common difference between the simple staining and gram staining is to know about the morphology, whether it is the shape of bacteria exists in either cocci or bacilli or in the form of chains or in the form of spirillum. Stain is one of the important tool which is used in the microbiology laboratory. Before going to discuss about staining technique, every microbiology student must and should know about the smear preparation. What is a smear? A smear is nothing but a culture. So, before going to start the experiment, whatever the culture that should be processed before 24 hours. There are different types of staining techniques, especially simple staining and gram staining. In simple staining technique, the bacterial cell morphology, size, shape, etc. that should be observed in the simple staining technique. And here it is a single step process. The name itself, simple staining, is a single step process in which methylene blue is one of the stain which is commonly used in the simple staining technique, and observe the color of the bacteria. In the result interpretation, we should know about only violet color cells that should be observed under the microscope. The next staining technique is the gram staining, and this was given by Hans Christian Gram in the year 1884 to know the difference between the gram positive bacteria and gram negative bacteria. Especially the common difference between the gram positive and gram negative bacteria is mainly it varies in the cell wall composition. The cell wall composition of the gram positive bacteria is mainly made up of peptidoglycan layer, whereas the cell wall composition of gram negative bacteria is made up of lipopolysaccharides. That is the common difference in the gram staining technique. In this particular staining technique, first take a clean glass slide and add a drop of suspension. The basic requirements for the gram staining technique is, first one is the reagents. So, here there are 4 reagents. The name itself, it is a differential staining technique. That is, in this particular technique more than 2 stains are used in the process. The primary stain is the crystal violet, and the mordant is the Burke's iodine, and 95% ethyl alcohol, and counter stain is the safranin. These are the 4 common stains which are commonly used in the gram staining technique. Why we call it as a primary stain? Because at primary stain is the stain which is the first stain used for the reagent, used as the reagent. So, this particular staining technique is mainly used to differentiate gram positive and gram negative bacteria. In this particular staining technique, first take a clean glass slide and add a drop of bacterial suspension to the glass slide, and add 1 or 2 drops of this crystal violet stain, and kept that slide for 30 seconds. After 30 seconds, wash the slide under distilled water. And second stain is the mordant. This is also called as Gram's iodine or Burke's iodine, and this mordant is a fixing agent. The main purpose of this particular mordant is to fix the stain in both gram positive and gram negative bacteria in stable condition, and this mordant it is also called Burke's iodine. And third stain is the 95% ethyl alcohol, and fourth stain is the counter stain. After washing the slide with this distilled water, add 95% ethyl alcohol to the bacterial smear, and finally counter stain. Why we called it as counter stain? Because it is the last stain which is used for this particular staining technique. So, that is the counter stain. And if you come to the interpretation of the result, gram positive appears in one color, gram negative appears in another color. So, gram positive appears in purple color, gram negative appears in pink color if you observe it under the microscope. The common mechanism inside in this one is because of the cell wall composition of gram positive and gram negative bacteria, the cell wall composition of gram positive bacteria is made up of peptidoglycan layer, and it is a very thick layer. Peptidoglycan layer is very thick layer which does not allow the other strains inside the cell itself. And the second gram negative bacteria, it is made up of lipopolysaccharides. Especially if you come to the composition of the lipopolysaccharides, these lipids are easily soluble in alcohol content. So, if you compare this result in the first step, both the gram positive and gram negative bacteria appears in purple color. In the second step, if you add mordant, especially this mordant acts as the fixing agent, that is, it can completely fixes the strain in both gram positive and gram negative in purple condition. And if you add the counter strain 95% ethyl alcohol, this 95% ethyl alcohol is very important and this is the crucial step in the gram staining technique. Especially this 95%, if you treat the slide with or if you treat the bacterial suspension with 95% ethyl alcohol, this gram positive, because of the peptidoglycan layer, it does not allow the ethyl alcohol inside the cell itself, whereas the gram negative is made up of lipopolysaccharides, that is the cell wall composition. So, this gram negative completely, this 95% ethyl alcohol wash the lipid layer such that the lipid layer is completely removed from the gram negative cell wall. And in this particular step, if you observe it under the microscope, gram positive appears in purple color, whereas the gram negative appears in this colorless colonies. And finally, if you treat with counter stains such as safranin, both already gram positive is having crystal violet inside itself, whereas the gram negative is the cell wall composition is colorless. So, if you treat with safranin, this safranin is completely absorbed by gram negative cell wall. Whereas, if you observe this gram positive cell wall, already crystal violet is present in the inside the cell wall, and once one stain is present inside the cell wall, the cell does not take the another stain inside itself. So, gram positive remains purple in color, whereas the gram negative is colorless. Already the stain is washed in the gram negative cell wall. So, this gram negative completely take the this particular safranin and observe the safranin. In this particular step, if you observe this particular slide, gram positive appears in purple, whereas the gram negative appears in pink color. This is the common difference between the gram positive and gram negative cell wall composition. This is the common difference, and especially in simple staining technique, only one stain is used in the simple staining technique, whereas the gram staining is more than 2 stains are used in the gram staining technique. So, that is why it is called as a differential staining. Why we call it as differential staining means more than 2 stains are used in the staining technique. This is called as a differential staining. And next and there are other types of stains also. One is the negative staining, especially this particular staining is mainly used for the capsulated bacteria. So, capsules, these are the protective layers that covers this particular microorganisms, especially this capsule that should be observed by this particular negative staining technique. And if you are coming to the microscopic observation, for the observation of this particular gram positive and gram negative bacteria, there are different types of microbes like simple microscopy, compound microscopy, phase contrast microscopy, fluorescent microscopy, and electron microscopy. And especially this is the normal bacteria which you can observe under the this particular compound microscope with 100x immersion objective lens. 100x immersion objective lens is called as cedar wood oil objective lens. So, this is about the common differences between the gram staining and simple staining technique. One technique is used for the morphology of the bacteria, and second technique is used to differentiate gram positive and gram negative bacteria. Under oil immersion microscope, we can observe the microorganisms. Gram positive appears in purple color, whereas the gram negative appears in pink or pink color. That is the common staining. These are the common staining techniques. And the important point here is smear preparation is also one of the important point. After the observation under the microscope, we observe different shapes of bacteria, like cocci. For example, if the bacteria appears in spherical form, it is called as a cocci. And for example, if you observe the bacteria in the form of rod shape, it is called as bacilli. Basing on the cocci, there are different shapes of cocci, that is streptococci. If the cocci is in the form of chains, it is called as a streptococci, whereas the staphylococci, if it is in the form of a bunch of grapes, if you observe the microorganisms in the bunch of grapes, it is called as staphylococci. That is those are the shapes of bacteria. And bacilli, like that monobacilli, streptobacilli, like that there are different types of bacilli. This is the shape of this particular bacilli is the rod shape. So these are the common differences. There is no specific arrangement in this particular staining technique. Only the main motto of this particular staining technique is to know about the morphology and to differentiate the bacteria in different forms. So this is about the bacterial culture staining using GAM staining is like a gram negative bacilli with no specific arrangement. So this is about the gram staining technique. Finally, I conclude this particular gram staining technique basing on the morphology. We can analyze the microorganisms by using this particular staining technique. We can analyze the microorganisms in different shapes. This is about the gram staining technique.